So our next session will be given by Louw, Claassens, and finally some staff sent me the printing so I can introduce Dr. Louw Claassens. So Dr. Claassens completed her PhD in marine biology in 2017 from Louw University, South Africa, and she's well known for her research and conservation work for the endangered Nisna seahorses. While still being a research associate at Louw University, she has recently joined the Palau International Coral Reef Center as a science officer and researcher for the Palau National Marine Sanctuary. Her keynote speak today will give us a review of the current knowledge of syngnathid within Southern and Western Africa, especially the critically endangered Australian pipefish and the endangered Nisna seahorse. Let's welcome Dr. Claassens.
Awesome. Thank you so much. Just to make sure everyone can hear me okay over there.
Yes, we can see you.
Okay, awesome. So thank you to the organizers for providing an opportunity for me to talk about how we are turning the tide on syngnathid conservation and research in Southern and Western Africa. This presentation actually came at a very opportune time, as a few colleagues and I have just completed a review of sygnathids for this region. So I'm going to start off with an overview of the Southern and Western African sygnathids. So we found around 63 syngnathid species within Southern and Western Africa, 11 seahorse species, 50 pipefish species, and then 2 pipehorse species, one of which is still being described at the moment. This represents 40% of the world's syngnathid genera. The highest number of species were found on the east coast of Africa, with high endemism in South Africa. So through this review, we looked at any and all publications that even remotely mentioned sygnathids. And when we plot that across the region, you can see a very distinct focus in the literature on the East Coast, and then specifically in South Africa. Then if you look at locality information in this research, publications that provide locality information of where sygnathids were found, you can see a similar pattern. A lot of focus on the East Coast, and then specifically in South Africa, specifically for the seahorses, and that's probably owing to Hippocampus capensis in South Africa. So looking at these two maps, you might think that syngnathid research in Africa is doing well, there's no problem, there's loads going on. But if you untangle and look at the different types of publications and research, you will find that a lot of it was species lists that looked at historic records when publishing the different syngnathid records. Some were management documents, reviews, and then species descriptions. Most of the ecological research publications that we found were not focused on sygnathids. They were general fish surveys or biodiversity surveys. And I think everyone tuned in today will appreciate that sygnathids require some sense of focus, especially in terms of how you survey them and how you look for them. Then we also turned to citizen science and iNaturalist. We developed a group called Sygnathids of Africa, where we collated all sygnathed observations in this region. And this really helped us to get a good idea of what divers are seeing on the ground, where they're seeing it. And it also provided a good opportunity to help with identification. Because again, I think everyone here can appreciate these guys are difficult to identify, especially from photos. So this group is still online, it's still open, and it provides a good idea of ongoing observations within the region. And then I need to touch base on probably the most exciting sygnathed news from Africa, probably ever. And that was the discovery of the first pygmy seahorse within Africa. So this little guy was discovered in 2017 by a local diver in Sudwana Bay. And it was only recently formally described by Graham Short, colleagues, and I in 2020. What makes this very special is that it was described in Sudwana Bay, a very popular diving site within South Africa. It's heavily dived, and even in such a popular area, there are still new species to discover. So it's kind of an indication of the potential of species discovery within the African region. Moving on, I want to dive a little bit deeper in 3 research projects that we have been involved in. And with each intro, you can see the different partners that have been involved and integral in developing this research and taking it further. So firstly, I want to focus on sygnathids from Mozambique. And I need to start at the place where this all came to be. It started in 2018 with a big confiscation of poached seahorses in the tiny town of Vilanculos in Mozambique. Various NGOs on the ground got in touch, and they wanted some assistance and help with how they can gather data and learn more about the poached seahorses that were collected to help with conservation efforts. And we gladly obliged and helped them to figure out what species were poached, how many were poached, as well as looking at some size data. And some initial data from that one poaching event in the small village was that 1,782 seahorses were confiscated. The majority of those, 88%, were Hippocampus camelopardalis, and then 13% of the dried poached seahorses had inflated brood pouches. The graph also gives a good idea of the different sizes of the seahorses. And this is important initial data looking at bycatch and even poached seahorses within a small village in Mozambique and what effect that might have on the local communities. But while we were busy with this, we realized we did not know anything about sygnathids in Mozambique. So we turned, one initiative that was developed to try and alleviate the pressure of poaching from the local community, because it's a big form of income to the local community, was developing seahorse tourism. And in the picture, you can see past poachers of seahorses turned seahorse guides. And these guys, with the help from a local NGO, is providing seahorse tours to tourists in the area as a means to generate alternative income. So the pressure to poach seahorses for income is now alleviated by providing tourist opportunities and generating income in that way. So moving on, as we did this research, we discovered we didn't know a lot about sygnathids in the area. And we teamed up with 2 local researchers, Elidio and Amancio, and through generous funding from local organizations and local lodges like Pambela Lodge, these guys were upskilled to become scuba divers. And with the seahorse finder Amancio, we were able to conduct 2 discovery expeditions to Vilanculos to see what the diversity of sygnathids were like in this area as an initial look and understanding. And we were not disappointed. And I had to add in some sygnathed pictures, of course. The most common seahorse we found was Hippocampus camelopardalis. And it should be kept in mind that this species is data deficient on the IUCN Red List at the moment. So any data that we are gathering is important for the future conservation of this animal. We also found Hippocampus kuda, and then some other seahorses that were quite interesting that we are investigating further with the help of Graham Short and Nuno Monteiro. In addition to the seahorses, we also found various pipefish species, which we will also hopefully investigate further to confirm taxonomy and learn more about them. But discovery wasn't all that we wanted to do. We wanted to get an idea of trends and patterns in number of seahorses over time. And with the help of Amancio and Elidio, we set up a monthly monitoring program at 10 sites in the Bay of Vilanculos, as you can see here in the map. And for the past 10 months or so, these guys have been gathering seahorse data on a monthly basis. And you can see here, it's mostly Hippocampus camelopardalis and kuda They occur together at most of these sites, but Hippocampus camelopardalis is the most common species found. And the exciting thing is, as we learn more about the taxonomy and the species in this area, additional seahorse species might be added to this figure. So in 2020, tourism took a knock, as the rest of the world. And in January 2021, another haul of seahorses that were poached were confiscated. So we realized there needs to be other approaches in addition to seahorse tourism to try and fight against the poaching. So with the help of Juliet and Parco, the local NGO on the ground, we are looking at training up rangers that you can see in the photo that will be conducting patrols, that would monitor protected areas for seahorses. And what makes this wonderful is that our longer term monitoring, our monthly monitoring, will inform where these seahorse sites will be. So this research and project is ongoing, and hopefully with the next SynBio, we'll have some interesting species discovery research and some wonderful seahorse conservation successes that we can share. I want to jump all the way to South Africa now, to Hippocampus capensis, located in only 3 estuaries along the south coast of South Africa. So this species is one of only 2 seahorse species in the world listed as endangered, and it is specifically found only in estuaries and depends on seagrass and algal habitats. So the major threat to this species is habitat loss or alteration, and within the estuaries where it's found, it's specifically the nutrient inputs that is affecting the habitat where it lives. So past research that we've done focus on the impact of the development of a residential marina estate on the abundance, the distribution of the species in the Knysna estuary. And this development is built out of Reno matrices and gabions, which are essentially wire boxes filled with stones. And the past research that we've done found that the Knysna seahorse really enjoys the wire matrices of this habitat. We found that the seahorse readily chooses the artificial habitat over natural habitat when given a choice. And we also found significantly higher densities of seahorses within this artificial habitat. And from the photo, you can see it creates a very nice 3-dimensional habitat with a lot of crevices where a cryptic species can hide in. So moving forward, we turned to mark recapture research, where we used 5 tags, which are small elastomer tags that you can inject just under the skin of the seahorse. And that provides a unique numbered code for each seahorse. So you can, in effect, follow a specific seahorse for a period of time and find out more about population sizes, growth, and movement. So even though we were limited with the number of seahorses we were allowed to mark, as well as the number of sites we could focus on, we decided to pick one area within the residential marina estate where high densities of seahorses can be found. And we marked around 80 seahorses and followed them for a 13-month period. And this graph shows the resighting rate of seahorses in this study, which was quite high. Of the seahorses marked, 70% were resighted. The highest number of resighting for a specific seahorse was 17 times. So using the 5 tagging approach was quite effective to monitor the seahorses over time. We also found, when we conducted an open population assessment, a decrease in seahorse population size over time. Now, the reason for that is not that clear. A study like this should ideally continue for multiple years so that annual patterns can be investigated as well. Seahorse populations are known to increase and decrease, so it would have been nice to continue this for a little bit longer to try and untangle this decreasing pattern. We also looked at growth, and this species also showed rapid initial growth, similar to other seahorse species. This was also limited by the number of animals we could mark and the number of juveniles we could mark and also find again. When we looked at movement, we found that they do not move far, There were limited differences between males and females or throughout the day. And on average, for a 12, 13-month period, this animal only moved around 5 meters. So it's safe to say if it finds a spot of rhinomatris it really likes, it tends to stick around, which is quite important for the conservation of this species. Seeing that the movement in this artificial habitat is not far, it's important to make sure that there's no localized impacts to these areas. So looking at the bigger picture of the Knysna estuary, and I hope this video will work, we wanted to figure out what the habitat extent is like within the estuary. And to do that, we used an open ROV, a trident ROV, to map the subtidal habitat. So we put down the ROV, we classified and identified the habitat, and that allowed us to create a habitat map with the extent of subtidal habitat for the entire estuary. This provided us a good idea of the area of occupancy available to the species, and seeing that habitat loss and alteration is the major impact, this provides a critical baseline data set that can be monitored over time to see if habitat is maintained, if it's increased, or if we're losing it. And that would help us to determine if our conservation efforts are actually working. So ideally, this exercise should be expanded to the other 2 estuaries where the species is found. Moving on to Syngnathis watermayri, the most threatened species of Syngnathid in the world. And I had to step back a little bit and provide the background to this species. It has quite a tumultuous background. It was discovered in '63 with a historic range of only 3 estuaries. So since its discovery and through various surveys, no pipefish were found. And in '94, it was classified as extinct. But alas, during surveys between '93 and '96, 43 adult pipefish were found in another estuary further up the coast from its original range. And since then, the pipefish has been classified as critically endangered. And this is the little guy. It's very cryptic and very difficult to photograph. It's found only in estuaries, and it lives in dense eelgrass and macroalgal beds. So what is the current status of this species? At the moment, it is only found in 2 estuaries along the south coast of South Africa. It is locally extinct in 3 other estuaries. So all this information is pretty much based on past research, historic research that was not really focused on the species specifically. So in 2019, we got funding from the National Geographic Foundation, and we decided to put some effort into focused research on this species. You can see that we surveyed 23 sites respectively in both the Bushmans and the Kariga estuary. And we also surveyed the other 3 estuaries where the species is locally extinct at the moment to see if it might be there since it was last seen in 2001. So at each sample site, we conducted 3 SayNet surveys where you do 3 sweeps with a SayNet, but we also focused on habitat because similarly as Hippocampus capensis, this species depends on habitat for its survival. And for habitat, we identified the type of habitat, the percentage cover, and we also looked at length of the eelgrass. For water quality, we focused on temperature, dissolved oxygen, salinity, and pH. But the salinity was the important aspect. Historically, it was thought that a loss of freshwater inflow into these estuaries is the major threat to this species. So fresh water is lost from these estuaries because of impoundments, abstraction, drought, etc. The thinking is without freshwater inflow, this has a big impact on the prey animals this species feeds on, and that directly impacts the pipefish. So we wanted to look at salinity and see if it's found if there's high salinity, and then try and untangle if high salinities and a lack of fresh water is actually a major impact. So what we found in terms of habitat, we found very dense beds of eelgrass and the macroalgae codium, and distributed across both the Bushmans and the Kariga estuaries in pretty high densities. To give you an idea of what the habitat looks like, It's dense beds of eelgrass with the macroalgal codium lying at the bottom of the eelgrass beds. And this provides a nice three-dimensional complex habitat to this species. When we looked at salinity, we found pretty much seawater. The lowest salinity we found was 30 parts per thousand, where seawater is around 35, 36. And we also found hypersaline conditions of 44 parts per thousand. So it's safe to say that the habitats where this animal is found is pretty saline at the moment, with limited to no freshwater pulses or inflow at all. So you would expect not a lot of pipefish. But luckily, we actually found some pipefish. For our first survey in October 2019, we found 8 pipefish. In March 2020, we found 28 pipefish. And in July 2020, 23 pipefish. You can also see these guys are quite well distributed across the whole length of the estuaries. And the highest number of pipefish we found in one net sweep was 11, which is pretty high. It's the highest density for this animal that has ever been recorded. So when we looked at what drives the presence of the species, it was specifically habitat that had a positive effect on if the species occurs or not. And specifically, it was the codium habitat. Eelgrass also had a positive effect. And even though there was a negative effect of increasing salinity, this was not really significant. But now we have to consider if our sampling approach was actually the right one. Here you can see us seine netting in a massive bed of eelgrass and codium. So you would expect we are not very effective in finding this animal. So to answer this question, we turn to environmental DNA. And very quickly, eDNA is when you collect water samples in your habitat, you filter it, and then you extract the DNA within your samples. Then you use different primers or assays to try and detect if your species is in that sample you extracted. What's important here for Syngnathid watermeri is that we developed a species-specific primer. And for that, we teamed up with the Trend Lab from Perth and the PhD student over there, Georgia Nestor, who developed a species-specific primer. So we tested it out. Is eDNA an effective method to detect the species? Maybe better than seine netting? And to do that, we also collected eDNA samples during the October 2019 survey. And from this graph, it is pretty evident to see that eDNA is very effective in picking up this rare and cryptic species, significantly better than the conventional seine net surveys that we usually do. So considering this information, the fact might be that this species might occur in other estuaries along the coast. We were just not very good at finding it. So going forward, we would like to expand our eDNA and our seine netting surveys to additional surveys along the coast to see and investigate the range of this species. We also looked at the habitat extent within these two estuaries, and this again provides important baseline data, or not baseline, but monitoring baseline data where we can compare future habitat changes to. So considering all that information and moving forward, we identified a few research gaps and conservation opportunities, and I'm quickly going to run through it. I hope I'm not over time yet. The first is syngnathid exploration. There is so much scope and potential of understanding and learning more about the species we find in Southern and Western Africa. I am very confident there are various species out there that we still need to describe. And then species-specific research. We need to get out there and understand the ecology, the life history of specific species. And that does not mean going out and noting a syngnathid when you do a general fish survey. It means going out and spending hours in the water trying to find these cryptic and rare species. And then using different tools to do that, like eDNA. Then a focus on conservation through regional and national laws, regulations, and policy. Local governments need to firstly know what a syngnathid is, and then be informed about the importance of adding them into local laws and regulations, specifically as a flagship species to protect coastal habitats that are vulnerable, such as mangroves, seagrass, and coral reefs. Another important aspect is locally significant education and awareness, making it relevant to the people on the ground that live with the different sygnathids on their doorstep. A focus should be placed on poaching and illegal trade, specifically where it's relatively small scale in the greater scheme of things, as these poaching events can have quite a big impact on the local syngnathid populations. And then capacity development. With the next SyngBio, it'd be wonderful to have a whole African session with African syngnathid researchers providing findings from their research. As part of our review, we also developed a very nice schematic of the different species of sygnathids found in Africa across the different habitats they are found in. So that's from rivers, fresh water with different impacts and different conservation implications, all the way to deep coral reefs. And we also provided a quick snapshot of the specific impacts in these different habitats, as well as some of the conservation efforts that could be implemented in these different habitats. So hopefully that review will be available soon, and we can share this resource with African conservationists, but also with conservationists globally. And that is my spiel on African sygnathids. I'm providing my email address here if anyone would like to get in contact with any of the research we are doing within the region. Thank you.
Okay. Thank you, Louw, for your talk. And we can have a few questions for your section. Are there any questions here in the room here? Any question online? Maybe I can ask a question. And it's actually a question that was raised by one of our guests here in the room. And he wonders why you find like the male seahorses often have larger body size than the female seahorses in the wild. So what is the possible reason for this kind of difference?
Oh my gosh, I don't know if that's a question for me specifically. I don't think, if I understood the question correct, I don't think I can provide an answer for that. I'm sorry.
Yeah, it's a very curious question for me as well. I often wonder because the graph actually shows that the female seahorse have larger body size, right, than the male seahorse, and so it's kind of curious why they have different kind of body size, and
the why of it is interesting. A lot of my research finds that is what it's like. But then, of course, the additional question is, why is it like that? And that's where additional research and insights from other researchers would be very valuable.
Is that because, how about the sex ratio? Is that because there are more females than males in the wild? So the female will compete each other for the mating chance?
That might be a reason, especially for Hippocampus capensis. We did find a female bias sex ratio in some of the habitats. It also varies across habitats and across seasons.
Okay.
And I know it's horrible to say longer term monitoring is required, but in some instances, to really get an idea of the overarching patterns and not just a snapshot of a pattern, the longer term kind of monitoring is quite needed.
Yeah, it's a really interesting question we can explore later in the future. All right.